Hello everyone, I am Saumya Darshan. My qualifications are MSc B.Ed and I am a biology faculty. So it's been 5 years. I have been teaching to the NEET level students as well as I also deal with the board level students. So in today's class we are going to discuss about the process of microsporogenesis. Okay. So without making any sort of delay, let's start our topic. Okay, so the process of microsporogenesis. In this process, we are going to discuss how a microspore is produced or a spore or the pollen grain that is being produced. How the spore is being produced. The spore is being produced. Is produced. Okay, so first of all, we have to know what are the male spore producing organ of a flower. If we talk about a flower, we can observe structures like this. This is basically the ovary part. This is basically the ovary part. Along with the ovary, as you know, there are three kinds of ovaries: are there hypogynous ovary, perigynous ovary, and epigynous ovary. Okay, superior ovary, half inferior ovary, and inferior ovary. Doesn't matter what is the size of the ovary, what is the position of the ovary. If the flower is a bisexual flower, that means it's also having male sex organs which are known as the androecium. In androecium, we can observe structures like this. Here, the structures which we are observing, these are known as filaments, and these are known as anthers. Okay, so togetherly we can say this is the male sex organ, or this is known as the androecium part of the flower. Okay, so right now I'm going to erase and typically going to focus on this structure, because from here only we can observe the microspore. Okay, let's draw a filament first. If this is a filament, two lobes are attached here. Okay, so lobe number 1 and a lobe number 2. A stuck like filament, it is attached with these lobes. These lobes, if we enlarge these lobes, we can see this will be look like this. If you are enlarging this, this is looking like this. This is looking like this. Here we can observe 4 chambers. Let's say this is the chamber number 1, this is the chamber number 2, chamber number 1, chamber number 2, chamber number 3, and chamber number 4. These chambers are known as theca. The chambers, these are known as theca. So we can say an anther is a bilobed. We can say an anther is a bilobed. Lobe number 1, lobe number 2, and tetrathecus. So anther are bilobed and tetrathecus. Okay, because we are observing 4 thecas. Okay, now let's enlarge this for a better understanding. Okay, if we enlarge this, we can see a structure like this. Okay, so here this is the chamber number 1, chamber number 2, chamber number 3, and chamber number 4. Here I can show you 4 lobe like structures, like rounded structures. These are known as, this is the just a large view of this. Okay, so these are known as microsporangium. These are known as microsporangium. Inside this microsporangium, we can observe certain kind of tissues. Okay. We can observe certain kind of tissues. These tissues are known as sporogenous tissues. sporogenous tissues. This sporogenous tissue is having a ploidy level of 2L. 2L. Okay, so let's say if a male is having a chromosome number of 30, so that means the sporogenous tissue is having its chromosome number of 30 only. But when it will produce spores, it will be reduced to haploid condition. That means up to 50. So I'm just giving an example. It's not like the exact data I'm giving. Okay. So now I'm going to erase this and going to draw a more detailed structure. If I draw like this, first let's say I'm drawing the microsporangium. Just above the microsporangium, I'll observe some layers. Okay, I'm going to observe some layers. The first layer which I will observe just like above the microsporangium, this layer is known as tapetum. Tapetum, that means it is the closest layer. This is the closest layer of the microsporangium. It might be uninucleated or it be binucleated. In major cases we have seen these are the binucleated cells. Okay, so above the tapetum I can observe some multilayer cells. Let's say these are the multilayer cells I am observing here. So multilayer cells I will be observing here. This multilayer cells will be known as middle layer. Okay. Now above the middle layer I will observe another layer of cells. This layer of cells will be known as endothelial cells. Endothelial. Just below the surface, that means this is going to be the last one. So this is going to be the last one. So the last layer I am drawing is known as the epidermal layer. Epidermal layer. So I can say a microsporangium which is containing sporogenous tissues which are diploid in ploidy level is having 4 layers. These are EEMT. EEMT, isn't it? E for epidermis, E for endothelial, M for middle layer and T for tapetum. Now let's talk about individual this sporogenous cells. Okay, so I am just talking about this. When the microsporogenesis process starts, it will be changed into this structure. Here I can clearly say these are in a stage of a tetrad. 1, 2, 3, 4. This is known as microspore tetrad. Microspore tetrad. But if we talk about how this tetrad came into existence, then first of all we have to know this was of 2N level. That means a first meiosis or meiosis 1 would have taken place. Meiosis 1 would have taken place. Meiosis 1 would have reduced its ploidy level, that to be 2N, that to be N. This one is N, this one is N. After a few time it would have completed the meiosis 2. As we know, meiosis 2 is equal to mitosis. So it might have changed into this kind of structures. Okay. So after few days or after few months, it depends upon the plant variety, this microspore tetrad will be seen like this. These are known as the pollen grains. If we talk about a pollen grain, these are the pollen grains, each are having n number of ploidy level. N number of ploidy level. Okay. So if we are talking about this pollen grain, it is having an outer covering, and this is going to be the outer covering part, and this is the outer one, this is the inner one, and here lies our N. Okay. So outer part is composed of a typical, a complex chemical whose name is sporopollenin. Sporopollenin. This is known as the exine part. The inner layer is composed of cellulose plus pectin, and this part is known as the intine part. Okay, so I can say the sporopollenin is going to be the hardest substance ever known. That means it will not be degraded by any kind of chemicals. Okay, so here I can also observe some germ pores will be here. Through these germ pores, our germ cell will come out, or our the pollen grain that will be come out. Okay, so if we talk about the pollen grain, it would be looking like something like this. It will be looking like something like this. That means it is having N or haploid. After few time, it will make another mitosis, and we can observe two cells lying in a like this one. This will be a, that means mitosis happened. Mitosis happened. That means this is a cell and this is a cell, but these are not truly differentiated. This cell, which is having a larger cytoplasmic mass, will be known as vegetative cell, and this will be known as generative cell. When they will be entering into the ovary, it will form a pollen tube. It will form a pollen tube. Major function of this vegetative nucleus is going to, along with its cytoplasmic mass, it is going to form the pollen tube. The generative nucleus will further go into the process of mitosis, and it will form another N here. One of these N will fertilize with the egg nucleus, and another N is going to fertilize with the polar nuclei. Okay, sorry, one will be with the polar nuclei, and yeah, one will be with the polar nuclei, and another one will be going to fertilize with the real egg nucleus. Okay, so this kind of fertilization is known as double fertilization. And if the polar nuclei, if the polar nuclei is fertilizing with this, that means it is having polar nuclei like this. Okay, okay, these are the polar nuclei. So the polar nuclei, if this is going to fertilize with this is a kind of fertilization, and egg nucleus, this is going to be fertilization. So two fertilizations are happening here. This will be known as double fertilization, and the fertilization is of a triple fusion. This will be known as a triple fusion. Okay. So here I conclude my process of like microsporogenesis. Here I can observe first one meiosis happened, then meiosis 2 happened, then again mitosis happened here, and that means here I am observing one meiosis and two mitosis. After that, I will be getting our male germ cell. Okay, so I hope you have understood this concept quite well and would have liked it. Okay, thank you.